Barbara McClintock, born on June 16, 1902, in Hartford, Connecticut, was an American scientist and cytogeneticist who has contributed to the advancements of our understanding of genetics. It all began in high school when McClintock discovered her interest for science and was dedicated to attend and continue her studies at Cornell University's College of Agriculture. Her wish came true and she received a PhD in botany from Cornell in 1927. From here, she would begin a study that she would devote the most time into and shape the rest of her career in life. I'm sure some of you know what I'm talking about, but for the people who don't, I'm referring to the development of maize cytogenetics. During her time at Cornell, she was interested in how to visualize chromosomes in mazes. McClintock researched the reasons for and how genes and chromosomes were able to shift and move during the breeding process of mazes. While scientists before McClintock studied crossing over, she built off that research and founded the physical basis of genetic recombination and crossing over. Crossing over is the process that takes place during meiosis, in which paired homologous chromosomes are able to partially exchange their genes with one another. Now you may be wondering, how did she figure all this out by just using some corn? Well, she created a new method that used an acetylcarmine staining process, which allowed her to see how recombined genes and chromosomes were related to the physical traits of maize. An example being color variations in the kernels. Furthermore, this technique allowed McClintock to create the first ever genetic map for maize in 1931 and laid the groundwork for connecting chromosomal changes to the physical traits of an organism. In her research, she also demonstrated the role of the centromere and telomere. McClintock throughout her career has won many notable awards, some of them being in 1944 when she became the third woman elected to the National Academy of Sciences, and in 1970 when she became the first woman to be awarded the National Medal of Science. She also won the Nobel Prize for her work on gene transposition and the mechanisms of gene expression in 1983. She's actually best known for her discovery of genetic transposition, or known as jumping genes, or transposable elements, which are DNA sequences that move from one location on the genome to another. For the majority of the 20th century, the agreed upon concept of genes was that they were some sort of stable entities arranged in an orderly linear pattern on chromosomes. However, that idea was challenged by McClintock's discovery of transposition. She used this discovery to demonstrate that genes have the ability to turn other genes on and off. Furthermore, she used the discovery of gene regulation to explain how organisms with identical genomes have cells that function differently. Fun fact about McClintock is that while she was in Cornell, she took up music and played tenor ukulele in a jazz band, even playing a few gigs around Ithaca, New York. She passed away on September 2nd, 1992, at the age of 90 due to natural causes. McClintock lived a full and great life, defying all odds and even breaking through the boundaries of gender during this time. She made advanced and significant discoveries that have forever shaped biology and the way we view genetics today. That's all, biologists. Make sure to review the video carefully and complete the study guide, as this will be on next week's Unit 6 test. Feel free to email me with any further questions.